Okay, right now let's talk about proteins. Right, proteins is another one of our organic or carbon-based molecules. So let's start off with, the thing that usually helps us out the most, which are going to be the elements for proteins. Elements for proteins, the one element that makes everything organic, that's carbon. And then the other two elements that we always see are hydrogen and oxygen. And then the two that we haven't seen yet in our macromolecules is N for nitrogen. And then sometimes we can see, let's just do that again. Sometimes we can see S, which is sulfur. Say sometimes, he's not in all of them. All right, now that I know my elements, let's do my monomer. So my monomer of a protein is an amino acid. And now that I know it's an amino acid, we can use our functional group knowledge to start to build this thing. Amino, so think of your functional groups. The functional group for amino is amine. And amine is NH2 versus my acid part. We only had one functional group that was acidic, and that was carboxyl or carboxylic acid. Carboxylic acid, and that is COO or COOH. So now all I have to do is build this amino acid. I know it has an amine group, an NH2, so let's start over here. So here's NH2, because I have two H's. Now what he attaches to is the one thing that makes this organic, which is my carbon. So I'm going to put carbon right here. I'm going to put a nice little symbol like that. That symbol, I'm going to put it over here. So that symbol means alpha. And the alpha, that means he's at the center of this entire thing. So I have carbon right there. He is really, really important. And then coming off my carbon, I'm going to have a hydrogen. And we'll talk about what comes off that other one. Now remember, carbon can form 4 bonds. So now I get to do my carboxylic acid. So here's my COOH. All right, so I have an amine group, which is right here. So this is my amino, and then my carboxylic is acidic, so amino acid. Now right here, we talked about how carbon has 4 bonds. He has 1 to my amine group, 1 to a hydrogen, 1 to a carboxylic group. Now this one down here is where he has an R group. And let's talk about R groups over here. An R group is going to be a functional group, which will, let's do this, which will determine the amino acids characteristics. And what I mean by characteristics right here is that sometimes these R groups can be hydrophilic, right? Sometimes they can be hydrophobic. Sometimes they can have a positive charge. Sometimes they can have a negative charge. Sometimes they are nonpolar and polar, say kind of the list goes on. And we'll need to know these characteristics to help us determine how a protein folds later on. But first, let's start building a protein using these amino acids. Now, if I want to build something, we've covered this before. If I want to build something, this is dehydration synthesis. Dehydration synthesis, build it. So let's take an amino acid. Let's take a nice easy one. So let's do NH2. So here's NH2. Attach that to my alpha carbon with a hydrogen with an R group, and then attach that to carboxylic acid. There you go. I have one amino acid right there. Now what I want to do is add another amino acid. So let's just draw a second one. So here's my NH2. Attach that to alpha carbon, hydrogen, R group. Attach that to carboxylic acid. And so now all I want to do is join these two things together. And we've talked about how we join things together with dehydration synthesis is by removing water. I dehydrate it. Well, if I'm looking at this amino acid right here and this amino acid right here, where they butt up against each other, I can see a natural water because I have a hydroxyl group and a hydrogen. If I combine those, I have H2O. That's where the water is. And so now that I found my water, you know that those bonds break apart. So the hydroxyl group is gone and that hydrogen is gone. And this carbon is going to attach to that nitrogen right there. So now we're just going to redraw this whole thing. And I'll even use the same color so you guys kind of see where they aligned up. So here's my NH2, alpha carbon, hydrogen, R. And then here's the C from the carboxyl. But now, since that OH is gone right here, I'm going to take this carbon and attach it to that nitrogen. So now, here's my nitrogen. Still has an H, alpha carbon, H, R, and then attach that to carboxylic acid. So now I have two amino acids joined together. This right here, this bond is really, really special. When I join two or more amino acids, because I could keep going, when I do two or more amino acids together, this is called a peptide bond. Okay, so when you have a peptide bond, when you have two or more amino acids joined together, this is called a polypeptide chain. And we know word roots. Poly means many. Peptide is referring to the bond of the amino acids, and that will form a chain. And so I can do that for hundreds and hundreds of amino acids, just keep adding on by doing dehydration synthesis. Now when I do that, I actually start building the structures of a protein, because proteins have four main structures. Okay, proteins have four main structures. The first one is called a primary structure. Now, a primary structure, by definition, is just going to be the chain of amino acids linked by peptide bonds. So what we drew up here, right here, this would be a primary structure because it's just a chain of them. I could just keep, oh, down here. So I could just keep doing this over and over and over again, be one straight line. If I made that picture a little easier, like this would be an amino acid, linked together, amino acid, linked together, amino acid, linked together, amino acid, etc., etc., etc. And then these bonds that are holding them together, those are going to be the peptide bonds. All right. At this point in an amino proteins, whatever you want to call it, life, this is not a functional protein when you're just a chain of amino acids. But now that you have your chain of amino acids, you know, we've talked a lot before that form is always function. So if something is shaped a certain way, it will do a certain job. This protein doesn't have a shape yet. He's just a chain of amino acids, so I have to start shaping him. The next structure, after I make a primary structure, is called a secondary structure. Now, a secondary structure, by definition, is going to be taking that primary structure and folding it two different ways. So my primary structure, which we usually use a 1 and a 0, so primary is 1, 0, secondary would be 2, 0. So secondary structure is when I take my primary structure and do two things. I can do one of two things. The first thing I can do are create alpha helices, or I can do beta pleated sheets. And what those look like, so an alpha helices, right? A helix, that's just when it corkscrews. So I can take this amino acid chain and I can start making that thing corkscrew. Or I can take the beta pleated sheets and do this with them. If you think beta pleats, have you guys ever folded a piece of paper to make it look like a fan? Like you just fold it back and forth on top of each other? That's what beta pleated sheets look like. So I took this one long amino acid chain. So there's my amino acid chain. That's my primary structure. When I make it into a secondary structure, it'll start to coil and it'll start to pleat. I can do it on the same amino acid chain, but now that's a secondary structure. At this point in time, still not a functional protein because I'm just beginning to do this. To hold these two structures together, to hold the beta pleated sheets and the alpha helices, these are held together, held by hydrogen bonds, okay? Held together by hydrogen bonds. Now, after secondary, you guys can probably guess the next structure. That's a 3. This is called, whoops, spelled it wrong. This is tertiary structure. And tertiary structure, again, with a 3 with a little 0, tertiary structure is going to be, it is made by the R group interactions. Because if I look up here and I look at this polypeptide chain right here, you can see how these R groups are hanging off the end. There's an R group and there's an R group. When I start getting a bunch of them together and I either start to coil or pleat it up, that's going to force them to get close to each other. And determining on what kind of characteristic your R groups have will determine how they interact with everybody around them. Because let's say we have charges. If I have a positive R group next to a positive R group, what happens? Well, think about magnets. When I put two positives together, they repel each other. So if I have two amino acids that are close that have the same charge, they're going to push away from each other. What about water? Like if they're hydrophobic, like they're afraid of water, and I put them next to something, right, I put them in the protein that's hydrophilic, right? Well, what that'll do, because remember most of our body is aqueous, right? So if I start folding this thing up, this is now a protein, right? If my hydrophobic ones, right, hydrophobic's afraid of water, that means they're going to start hiding in the middle of my tertiary structure because they don't want to be around the water. Versus maybe hydrophilic ones, these love water. So they're going to start to orient themselves on the outside. And that's when you start to get this tertiary shape. This tertiary shape is when it becomes, not a very fancy word, but it's when it becomes globular, when it starts to do this because of the interactions. And so maybe this was a positive charge, and so another positive charge, he's going to put himself, you know, as far away as he can from him because they don't want to be next to each other. But maybe what if I have a positive charge next to a negative charge? Well, that means they're going to want to be next to each other. So maybe here's a negative charge. So he's going to want to be close to that guy because that guy was positive. And that's when you start to get this really, really cool glob structure because of the interactions of the R groups and how they and where they want to be. Now for quaternary structure, so we have our last one. Oh, right up here, tertiary. This is when it becomes functional because I have my final shape. Now fourth, this is quaternary structure, which is a 4 with a little 0. Quaternary structure, what that looks like or what it is when you have one or more tertiary structures combined. And all that is like, here's a tertiary structure, but then to get a certain job done, maybe he needs to be hooked up with this other tertiary structure. This is now quaternary. Maybe I even have another one. Maybe I have 3 tertiary structures to make that. A really famous one, a really famous quaternary structure is a red blood cell, because he looks a lot like that. He looks kind of like a donut with a dip in the middle. And this is where, if you know red blood cells and what they do, they carry oxygen. Well, they carry gases, not just oxygen. They carry gases around our body. And so these gases, like oxygen, can sit here in this nice little dip where he's safe. Well, that dip is created because of the different tertiary structures combined. And he makes that little dip, and then he's able to carry everything all around. Last thing, for proteins, we need to do the function, okay? The function of proteins is to build a physical organism. That's all I'm doing with the protein, which I say all like it's nothing. That's a lot. But everything physical that you see about something living is dictated by a protein. So we could do really easy examples like eye color is dictated by a protein, or maybe how tall you are is dictated by a protein, or the fact if you have freckles or not, that's dictated by a protein, or if you have some type of genetic disorder, that's dictated by a protein. Maybe heart disease runs in your family, that's dictated by a protein. So anything that is made by your body is a protein, everything. Now there's a couple special proteins, right? The first one being, I kind of lost my train of thought. Build physical organisms. Yeah, oh, okay, the one special case, these are called enzymes. Enzymes, what they do, they help chemical reactions inside our bodies. But we'll talk more about enzymes just on another day. But I think this is everything you guys need to know for proteins. We have elements, we have monomers. We talked about how we build a polypeptide chain. And then once we have that polypeptide chain, the different structures it takes. And then what proteins do. All right, you guys did good.